Good evening. We're back with the continuation of the morning lecture, and we were at the topic of molecular chaperones oversee protein folding, which starts now. Welcome, all of you. Welcome back to New Age Cosmetic White EdTech Healthcare Consultancy's regular classes for AIIMS&NEET Biology, NEET exams in biology and chemistry, and the Council for Scientific Industrial Research. For the national eligibility tests. So here we start on with the topic. I repeat, molecular chaperones oversee protein folding. Molecular chaperones or chaperonins are proteins that oversee the correct folding of other proteins. Many chaperonins belong to the family of heat shock proteins as their levels increase at high temperature. See, as in Chapter 16, regulation of transcription in prokaryotes. Chaperonins may be divided into two classes, holders and folders. I repeat, the chaperonins may be divided into two main classes, holders and folders, respectively, those that prevent premature folding and those that attempt to rectify misfolding. Obviously, chaperonins cannot know the correct 3D structure for several other proteins. Mechanistically, they act to prevent incorrect folding rather than actively creating a correct structure. And just to repeat the start of the paragraph, molecular chaperones or chaperonins are proteins that oversee the correct folding of other proteins. Many chaperonins belong to the family of heat shock proteins as their levels increase at high temperature. See Chapter 16, where we talk of regulation of transcription in prokaryotes. Now, on to the next paragraph. During bacterial protein export, the secretory chaperonin section B keeps the polypeptide chain from folding up prematurely. Secreted proteins must travel through a narrow translocase channel and so must remain unfolded until they reach the other side of the membrane. The HSP70 of chaperonins tends to bind to new synthesized or highly uncoiled proteins. The more complex GRO-E, that's HSP60 by HSP10 chaperonin machine, attempts to refold damaged or misfolded proteins. When polypeptide chains unfold, they expose hydrophobic regions that are normally clustered in the center of the folded protein. Left to themselves, many proteins could refold. However, inside a cell there is a high concentration of protein. Consequently, exposed hydrophobic regions from multiple proteins bind to each other and their proteins aggregate together. The GRO-E chaperonin machine forms a cavity in which a single polypeptide can refold on its own, protected from interactions with other polypeptide chains. Newly made proteins face a folding problem. The N-terminus of the growing polypeptide has already left the ribosome, while the C-terminal region is still being made. Consequently, the N-terminal region does not yet have access to any folding information that resides in later regions of the protein. To prevent misfolding at this stage, the emerging protein is sheltered by a chaperonin known as trigger factor. This binds to the large subunit of the ribosome close to the polypeptide exit tunnel. Protein synthesis occurs in mitochondria and chloroplasts. Mitochondria and chloroplasts are thought to be of prokaryotic origin. The symbiotic hypothesis of organelle origins argues that symbiotic prokaryotes evolved into organelles by specializing in energy production and progressively losing their genetic independence, as seen in or explained in Chapter 4 on genes, genomes, and DNA for the details. Both mitochondria and chloroplasts contain circular DNA that encodes some of their own genes and they divide by binary fission. They contain their own ribosomes and make some of their own proteins. Organelle ribosomes, resemble the ribosomes of bacteria rather than the ribosomes of the eukaryotic cytoplasm, box number 1303. The initiation and elongation factors of organelles are also bacterial in nature. And now for the terms of importance. Chaperon is sometimes molecular chaperone, same as chaperonin. Chaperonin is a protein that oversees the correct folding of other proteins. Heat shock protein is HSP protein induced in response to high temperature. Many heat shock proteins are chaperonins. And now on to figure 13.5. The chaperonins act by two general mechanisms. A. Chaperonins of the HSP70 type act during protein formation by binding to hydrophobic patches of the protein. Once chaperonins are released, the protein automatically folds. B. Large chaperonins such as KORAH-E act after translation by sequestering misfolded protein in a central cavity. Freed from the influences of other molecules in the cytoplasm, the protein will fold correctly. And the side note reads that protein synthesis in mitochondria and chloroplasts resembles that of bacteria in many respects. And the topic says that there are differences in composition between organelle and bacterial ribosomes as shown in 1306. And the next topic, 12.1, that's proteins are imported into mitochondria and chloroplasts by translocases. And the size of organelle genomes varies considerably from organism to organism. Generally, the more advanced eukaryotes, right, and proteins are imported into mitochondria and chloroplasts by translocases. The size of organelle genomes varies considerably from organism to organism. Generally, the more advanced eukaryotes have smaller organelle genomes. The mitochondria of mammals make only around 10 proteins, and in higher plants, the chloroplasts make approximately 50 proteins. The other organelle proteins are encoded by nucleogenes and made on the cytoplasmic ribosomes. They are then transported into the organelles. The more complex KORAH-E, which is equal to HSP60 by HSP10 chaperon machine, attempts to refold damaged or misfolded proteins. When polypeptide chains unfold, they expose hydrophobic regions that are normally clustered in the center of the folded protein. Left to themselves, many proteins could refold. However, inside a cell there is a high concentration of protein. Consequently, exposed hydrophobic regions from the multiple proteins bind to each other, and proteins aggregate together. The KORAH-E chaperone machine forms a cavity in which a single polypeptide can refold on its own, protected from interactions with the other polypeptide chains. Newly made proteins face a folding problem. The N-terminus of the growing polypeptide has already left the ribosome, while the C-terminal region is still being made. Consequently, the N-terminal region does not yet have access to any folding information that resides in later regions of the proteins. To prevent misfolding at this stage, the emerging protein is sheltered by a chaperone known as trigger factor. This binds to the large subunit of the ribosome close to the polypeptide exit tunnel. Protein synthesis occurs in mitochondria and chloroplasts. Mitochondria and chloroplasts are thought to be a part of the prokaryotic origin. The symbiotic hypothesis of organelle origins argues organelle ribosomes resemble the ribosomes of bacteria rather than the ribosomes of the eukaryotic cytoplasm. The initiation and elongation factors of organelles are also bacterial in nature. Now for the terms of importance. Chaperone, sometimes molecular chaperone, same as chaperonin. Chaperonin, protein that oversees the correct folding of other proteins. Heat shock protein, HSP protein induced in response to high temperature. Many heat shock proteins are chaperonins. Now for explanation on the figure shown here, 13.35, chaperonins act by two general mechanisms. Chaperones of the HSP70 type act during protein formation by binding to hydrophobic patches of the protein. Once chaperones are released, the protein automatically folds. B. Large chaperones such as KORAH-E act after translation by sequestering misfolded proteins in a central cavity. Freed from the influences of other molecules in the cytoplasm, the protein will fold correctly. And there are differences in the composition between organelles and bacterial ribosomes as shown. Protein synthesis in mitochondria and chloroplasts resembles that of bacteria in many respects, is the side note. Proteins are imported into mitochondria and chloroplast by translocases. The size of organelle genomes varies considerably from organism to organism. Generally, the more advanced eukaryotes have smaller organelle genomes. The mitochondrial mammals make only around 10 proteins, and in higher plants, the chloroplasts make approximately 50 proteins. The other organelle proteins are encoded by nuclear genes and made on cytoplasmic ribosomes. They are then transported into the organelles. Proteins for import into mitochondria have a leader sequence at the N-terminus. This consists of 20 or more amino acids with a positively charged lysine or arginine, every 3 or 4 residues, and no negatively charged residues. The leader forms an alpha helix with a positively charged phase and a hydrophobic phase. This is recognized by a receptor on the mitochondrial surface. The protein is imported successively through 2 translocase complexes known as TOM, that's translocase outer mitochondrial, and TIM, translocase inner mitochondria, that lie in the outer and inner membranes of the mitochondria respectively. After importing the protein, its leader sequence is trimmed off. Side note states that many mitochondrial and chloroplast proteins are made in the eukaryotic cytoplasm and enter the organelle after synthesis, as noted on the side note. Plant cells are more complex than animal cells, as they possess not only mitochondria but also chloroplasts. The principle of protein import is similar. The chloroplast contains 2 translocases equivalent to TIM and TOM, which are known as TIC and TOC, C for chloroplast. So there are leader sequences for chloroplast proteins. Next is on to figure 13.36, which is the trigger factor and the ribosome. And now, as you see, the figure 13.36 shows the trigger factor shelters newly made polypeptides as they emerge from ribosomes. Trigger factor protects about 70% of newly made proteins. The other 30% need the DNA-JK or GRO-EL chaperonins. Structure and function of the molecular chaperone trigger factor biochemical biofish, courtesy to all of them. Table 13.6 is about the components of cytoplasmic organelle and bacterial ribosomes. And as such, all these resemble those for mitochondria, and in fact, only plant cells can tell them apart. Thus, the mitochondria of fungi will import chloroplast proteins if genes encoding these are artificially introduced into the fungal cell. It is still unclear how plants decide between chloroplast and mitochondrial leader sequences. However, it seems that the leaders for the 2 kinds of organelles form different secondary structures. And the protein imports by organelles also need chaperonins on both sides of the membrane. An imported protein must travel through the narrow transclosed channel in an uncoiled conformation. To avoid premature folding, newly synthesized organelle proteins are kept in a loosely folded conformation by chaperonins. Later, when the imported protein emerges from the translocase into the inside of the organelle, it is bound by another set of these chaperonins. In particular, an HSP70 type chaperonin is responsible for hauling in the incoming protein. The HSP70 acts as a ratchet, binding to successive segments of unfolded polypeptide chain. Each binding and release of HSP70 consumes energy in the form of ATP. The figure 13.36 shows the trigger factor and the ribosomes. Trigger factors shelter newly made polypeptides as they emerge from the ribosome. Trigger factor protects about 70% of newly made proteins. The other 30% need DNA-JK or GRO-EL chaperonins. Such components of cytoplasmic organelle and bacterial ribosomes. Table 1306. 1303 hybrid ribosomes, just to mention that, although they are larger, the cytoplasmic nucleus encoded ribosomes of eukaryotes resemble those from archaea in the way they operate. A similar relation holds for the associated initiation and elongation factors. In fact, it is possible to make hybrid ribosomes containing one subunit from yeast and one from sulfolobus, an archaeon. These still make protein, albeit less efficiently than native ribosomes. In contrast, hybrid ribosomes made by mixing subunits from yeast and E. coli are totally functionless. So just going back to the previous thing, matter is taught to you. So the mitochondria of fungi will import chloroplast proteins of genes encoding these artificially introduced into the fungal cell. If these are introduced artificially, it is still unclear how plants decide between chloroplast and mitochondrial leader sequences. However, it seems that the leaders of two kinds of organelle form different secondary structures. Protein imported by organelles also need chaperones on both sides of the membrane. The imported protein must travel through the narrow translocase channel in an unfolded conformation. To avoid premature folding, newly synthesized organelles import proteins merges. Organelle proteins are kept in a loosely folded conformation by chaperones. Later, when the imported protein emerges from the translocase into the side of the organelle, it is bound by another set of chaperones. In particular, an HSP70 type chaperone is responsible for holding in the incoming protein. The HSP70 acts as a ratchet, binding to successive segments of unfolded polypeptide chain. Each binding on the release of HSP70 consumes energy in the form of ATP. Right. Continuing from where we left, we have the topic of where mistranslation usually results in mistakes in protein synthesis. Ribosomes are not perfect and make occasional mistakes. Perhaps 1 in 10,000 codons is misread and results in the wrong amino acid being incorporated. 2 amino acids whose codon differ by only one base are most likely to be confused. Other possible errors are shifts in the reading frame, that's frame shift, or reading through stop codons. These assorted errors are collectively known as mistranslation. Although such events are rare, a few weird genes actually require such errors for proper expression. For example, the pol gene of retrovirus, as in chapter 24, virus, viroids, and prions, is only translated if the ribosome frame shifts or reads through a stop codon while translating the preceding gag gene. So, as in chapter 10, cell division and DNA replication, it was noted that the DNA X gene of E. coli gives rise to 2 proteins, tau and gamma, both subunits of DNA polymerase. The gamma protein is made only as a result of frame shifting. Release factor 2, RF2 of E. coli also requires a frame shift for synthesis. The next is many antibiotics work by inhibiting protein synthesis. Many well-known antibiotics by inhibiting protein synthesis. Most of these are for specific prokaryotic ribosomes. However, very high concentration of these agents will inhibit the ribosomes of mitochondria and chloroplasts, which are of prokaryotic ancestry. Aminoglycoside antibiotics bind to the 30S subunit. Streptomycin binds to the 16S rRNA near where the 2 ribosomal subunits touch. The presence of streptomycin distorts the A site and hinders binding of incoming charged tRNA. In particular, binding of initiate initiator tRNA MET is inhibited, and so initiation of translation is prevented. Streptomycin-resistant mutants have alterations in nucleoside 523 of 16S-rRNA or in ribosomal protein S12 RPSL, which assists antibiotic binding. Many of the other aminoglycosides, such as gentamicin and kanamycin, bind to multiple sites on the 30S subunit, or mainly inhibit the translocation step of protein synthesis. Streptomycin and other aminoglycosides also cause misreading of the mRNA. Important words as we proceed. On the previous page, there will be 1. Aminoglycosides. Class of antibiotics that inhibits protein synthesis include streptomycin, neomycin, kanamycin, amikacin, and gentamicin. Frame shift. Alteration in the reading frame during polypeptide synthesis. Mistranslation is errors made during translation, and streptomycin will be an antibiotic of the aminoglycoside family that inhibits protein synthesis. The side note reads, tetracycline binds rRNA in the small subunit of both prokaryotic and eukaryotic ribosomes. Tetracycline inhibit both bacterial and eukaryotic ribosomes. They bind to the 16S or 18S rRNA of the small subunit and block the attachment of charged tRNA. Despite inhibiting both types of ribosomes, tetracyclines inhibit bacterial preferentially due to the fact that bacteria actively take them up, whereas eukaryotic cells export them. Chloramphenicol binds to the 50S subunit to a loop of 23S rRNA that interacts with the acceptor stem of the tRNA. And inhibits the peptidyl transferase. Cycloheximide binds to the 60S subunit of eukaryotic ribosomes and inhibits the peptidyl transferase. Erythromycin and related macrolides antibiotics bind to the 23S rRNA of the bacterial ribosome and inhibit the translocation step. Fusidic acid is a steroid derivative that binds to prokaryotic elongation factor EFG in the presence of fusidic acid. EFG, with its bound GDP, is frozen in place on the ribosome. Fusidic acid also inhibits the corresponding eukaryotic elongation factor EF2. However, in practice, animal cells are unaffected as they do not take up the antibiotic. Post-translational modification of proteins. Although the genetic code has codons for only 20 amino acids, many other amino acids are occasionally found in proteins. Apart from selenocysteine and pyrrolysine, discussed later, the extra amino acids are made by modifying genetically encoded amino acids after the polypeptide chain has been assembled. This is known as post-translational modification, as shown in figure 13.37 for a summary of some common post-translational modifications. An example of medical importance is diphthemide, which is derived from histidine by post-translational modification, figure 13.38. It is found only on elongation factor EF2 of eukaryotes and archaea, at a region of the amino acid sequence that is highly conserved. The corresponding bacterial factor EFG does not contain diphthemide. Diphthemide was named after diphtheria, an infectious disease caused by the bacterium Corynebacterium diphtheriae. Diphtheria toxin attaches an ADP ribose fragment to elongation factor EEF2 via diphthemide, and this inhibits protein synthesis and kills the target cells. EEF2 normally splits GDP and uses the energy released to move the peptidyl tRNA from the A side to the B side. ADP ribosylated EEF2 still binds GDP but cannot hydrolyze or translocate the peptidyl tRNA. Selenosystine and pyrrolysine, rare amino acids. Selenosystine sec is not one of the standard 20 amino acids, and yet it is incorporated into a few rare proteins during translation. During translation of the mRNA by the ribosome. This occurs both in bacteria and in eukaryotes, including humans. Sequencing of the genes and protein involved has shown that selenosystine is encoded by UGA. However, UGA is one of the stop codons. Apparently, UGA is normally read as stop but is occasionally translated to give selenosystine, which therefore has the honor of being the 21st genetically encoded amino acid. The choice between stop and selenosystine depends on a special recognition sequence in the following part. Before we proceed, let's just finish up the terms of importance. Chloramphenicol, an antibiotic that inhibits bacterial protein synthesis. Cycloheximide, an antibiotic that inhibits eukaryotic protein synthesis. Diphthemide, modified amino acid found only in eukaryotic elongation factor EEF2, that is a target for diphtheria toxin. Erythromycin, an antibiotic that inhibits bacterial protein synthesis. Fusidic acid, an antibiotic that inhibits protein synthesis. Post-translational modification of a protein or its constituent amino acids after translation is finished. Selenosystine sec, amino acid resembling cysteine but containing selenium instead of sulfur. Tetracycline family of antibiotics that inhibits protein synthesis. And the choice between stop and selenosystine depends on a special recognition sequence. In the following part of the gene, the selenosystine insertion requires cis element, SESIS element. Selenosystine has its own tRNA. I'm reading the side note. Very rarely the stop codon UGA is read as the unusual amino acid selenosystine. Selenosystine has its own tRNA and has a small protein initiator factor to escort charged tRNA sec to the ribosome. In fact, selenosystine tRNA is initially charged with serine, then the attached serine is enzymatically modified to form selenosystine. Selenosystine insertion sequence, SECIS element recognition sequence that signals for insertion of selenosystine at a UGA stop codon. Histidine and diphthemide. After the polypeptide chain of EEF2 has been made on the ribosome and one specific histidine residue is converted to diphthemide, diphtheria toxin stops protein synthesis by adding an ADP ribose group to the side chain of diphthemide. This inhibits the action of EEF2. Figure 13.39, delivery of tRNA with selenosystine to an internal guide UGA. Stop codon A. In bacteria, the tRNA carrying selenosystine sec first binds to cell B, and the complex then binds to a stem and loop in the mRNA. This aligns the tRNA sec with a UGA codon within the coding sequence on the mRNA. Selenosystine is then inserted as part of the growing polypeptide. Only the fully bound complex is shown B in mammals, the protein that binds the stem and the loop and loop and tRNA-seq is called EEF-seq. In addition to this, the stem and loop are more distant, being found after the stop codon. When bacteria and cysteine, the cysteine insertion sequence forms a stem, the loop structure in the mRNA molecule just after the UGA, cell B protein recognizes both charged tRNA-seq and this stem and loop. Thus, cysteine-bound tRNA is delivered to the right place, 13.39a. In bacteria, the stem and loop form temporarily from part of the coding sequence, and this section of the mRNA is therefore translated after insertion of the cysteine. In mammals, the stem and loop structure is found beyond the end of the coding sequence in the 3' untranslated region, not next to the central UGA codon. SEC-ish or SEC-ish binding protein 2 and the cysteine specific elongation factor EEF-seq are needed for binding the tRNA-seq and recognizing the SEC-ish stem and loop. The stem of the structure has a conserved sequence that forms a K-turn via a non-standard base pairing. This allows specific recognition. The tRNA-seq is then delivered to the correct position for insertion. Humans have about 25 genes that encode cellular proteins. These are divided into two groups of regulation. Some are expressed under all conditions, and others only when needed to combat oxidative stress. Selenocysteine is an analog of cysteine, but has selenium instead of sulfur. Selenium is more susceptible to oxidation than sulfur, and so proteins that contain it must be protected from oxygen. Examples of the formate dehydrogenases found in many bacteria. These contain selenocysteine in their active sites and function in anaerobic metabolism. They are inactivated by oxygen and are normally made up only in the absence of air. It has been suggested that the occurrence of cellular proteins in different groups of organisms is related to their oxygen sensitivity. Higher plants which make oxygen completely lack proteins that contain selenocysteine. Fungal genomes has also totally lack cellular proteins. Conversely, fish which live in the sea where oxygen levels are lower than on land have more cellular proteins. Figure 13.40, selenocysteine and cysteine. This is cysteine, this is selenocysteine. Right, selenocysteine is identical to cysteine except for the replacement of sulfur by selenium. Make a screenshot of this, which is important. Figure 13.41, pyrrolysine and lysine. Pyrrolysine is 5R, 4-methyl pyrroline carboxylate. So, continuing the subject matter purpose, this is note conversely, fish which live in the sea where oxygen levels are lower than on land have more cellular proteins than mammals. This is a continuation than typical mammals. Indeed, zebrafish cellular protein P contains 17 sec residues in the larger number in any known protein. In 2002, a 22nd genetically encoded amino acid was discovered, pyrrolysine, PYL, a derivative of lysine with an attached pyrroline ring. This is found in a few archaea where it is encoded by the stop codon UAG in occasional proteins. So basically we are under the impression that there are only 20 amino acids. The 21st happens to be selenocysteine and 22nd now is pyrrolysine. Right? This is found in a few archaea where it is encoded by the stop codon UAG in occasional proteins. Pyrrolysine was first discovered in the active site of methylamine methyltransferases found in methane-producing archaea of the genus Methanosarcina. An unusual aminoacyl-tRNA synthetase, a special tRNA, and genes for 3 accessory proteins are also found in organisms with pyrrolysine. By analogy with selenocysteine, it was thought that pyrrolysine tRNA was first charged with lysine, which was then modified to form pyrrolysine. However, this is proved to be wrong. Pyrrolysine is made first as a free amino acid and then attached to tRNA PYL. There is no pyrrolysine specific elongation factor. Moreover, the sequence that determines that specify which UAG codon should be used for pyrrolysine insertion are unclear. Genome sequencing has found genes homologous to those for the pyrrolysine system in occasional U bacteria, suggesting that pyrrolysine may be present. However, pyrrolysine itself has not been identified directly in these organisms. The fact that the same 5 genes in the same order are found in both archaea and eubacteria suggests that horizontal gene transfer has occurred. Next is degradation of proteins. Living cells not only synthesize proteins, they also degrade them. Although protein degradation is nowhere near as complex as synthesis, it is nonetheless carefully regulated and often highly specific. Proteases or proteinases are enzymes that degrade. Proteins, they are potentially dangerous to the organism that makes them and must be carefully controlled. Proteases are often located in separate compartments where they can act without endangering the components of the organism. Alternatively, proteases may be designed so that they only accept specifically tagged proteins for degradation. And going on to the terms of importance, like the terminologies. Protease, same as proteinase, an enzyme that degrades proteins. Pyrrolysine, PYL, it's 22nd genetically encoded amino acid derived from lysine. Digestive enzymes are activated on location. Proteases destined for export are made as precursors and are cleaved to form the active protease once safely outside the cell. B. Proteases in the membrane-bound lysosome degrade ingested material. Take a screenshot of this diagram. Continuing to the subject, proteases are found in 3 main locations: extracellular inside special compartments and free in the cytoplasm. Animals secrete proteases in their digestive tracts. These enzymes are usually synthesized as inactive precursors and only activated once they are safely outside the cells of the animal that made them. Figure 13.42a. Examples are trypsin and its precursor trypsinogen and pepsin and its precursor pepsinogen. Plants that catch insects, fungi that trap nematodes, and bacteria that live in rotting animal or plant tissue also secrete proteases. As with animals, these proteases are generally secreted as inactive precursors and only activated once outside the cells of the producer or organism. The side note reads, enzymes that degrade proteases are dangerous. They are frequently kept in separate compartments and often made as inactive precursors. Now, lysosomes are membrane-bound organelles found in eukaryotic cells. They contain a variety of digestive enzymes, including proteases, and function in self-defense. When cells of the immune system have engulfed bacteria or virus particles, the vesicles containing the invader are merged with lysosomes and the infectious agent is hopefully digested. Bacteria do not always cooperate. For example, many pathogenic strains of Salmonella can survive the toxins and digestive enzymes inside lysosomes. Proteases located in the cytoplasm itself must be very carefully controlled. Nonetheless, the cell needs some internal proteases to degrade damaged or misfolded proteins. Many proteases found inside bacterial cells form hollow cylinders with the dangerous active site on the inside of the cylinder. Proteins slated for destruction are ferried to the protease cylinder and pushed into its center by accessory proteins. The number of misfolded proteins and consequently the level of protein degradation increases greatly under certain conditions. In particular, when cells are exposed to uncomfortably high temperatures, they tend to disrupt protein structures. This induces the heat shock response described in more detail in Chapter 16, Regulation of Transcription in Prokaryotes. Eukaryotes have more sophisticated structures known as proteasomes. These are cylindrical, with the protease active sites inside. The top and bottom of the cylinder are covered by protein complexes that recognize and bind damage to unwanted proteins. Proteins destined for degradation are identified by tagging with ubiquitin. This is a small protein that is fixed to damaged or misfolded proteins, and also to certain proteins that are needed only for a certain period. Ubiquitin tagged proteins are unfolded and then fed into the barrel of the proteasome, where they are degraded into short peptides. The ubiquitin tags themselves are just cleaved off and recycled. A term of importance in the previous page is lysosome. Is a membrane-bound organelle of eukaryotic cells that contains degradative enzymes. Right, now proceeding to figure 13.43, operation of proteasome. Ubiquitin tags damaged proteins and is recognized by the cylindrical proteasome. After degradation, the polypeptide fragments and ubiquitin are extruded. Terms of importance, proteasome. Protein assembly found in eukaryotic cells that degrades proteins. Ubiquitin is small protein attached to the other protein as a signal that they should be degraded, used by eukaryotic cells, not bacteria. Right, now the key concepts. Polypeptide chains are linear polymers of amino acids. There are 20 common genetically encoded amino acids plus 2 rare ones. Right, and each amino acid is encoded by a codon consisting of 3 bases. Each codon is recognized by the corresponding anticodon on a transfer RNA tRNA to which the amino acid is attached. Translation is the synthesis of proteins using information from rRNA and is carried out by the ribosome. Codons can be read in 3 possible reading frames. Thus, it is important to locate the correct start codon by using recognition sequences. During elongation of the polypeptide chain, the tRNA moves between 3 separate sites on the ribosome. Translation is terminated when the stop codon is read by a protein known as release factor. The ribosome subunits are then recycled. Several ribosomes usually read the same mRNA simultaneously. In bacteria, a single mRNA may encode several proteins. In addition, since there is no nucleus, transcription and translation are coupled. There are differences between protein synthesis in eukaryotic and prokaryotic cells. Proteins to be exported from the cell have signal sequences that direct them to the export machinery. Molecular chaperones are specialized proteins that oversee the correct folding of other proteins. Some proteins are made inside mitochondria and chloroplasts, but most organelle proteins are made in cytoplasmic ribosomes and transported into the organelles. Many antibiotics inhibit protein synthesis. These include tetracycline and aminoglycosides such as streptomycin and chloramphenicol. The rare amino acids selenocysteine and paralysine are inserted at special stop codons. Proteins are broken down by proteases. In eukaryotes, proteins tagged with ubiquitin are degraded in cylindrical assemblies called proteasomes. Now these are review questions that you need to answer, right? What is a gene product? Are all gene products translated? Describe 3 gene products that are not translated. How common is the generalization 1 gene, 1 protein? True. Briefly give 3 examples of how 1 common gene can yield multiple proteins. Would you expect to find the same number of genes in the genomes as there are proteins in the proteome? Why or why not? Translate the following mRNA sequence into a protein. That's given below. And the next will be name 2 modified bases commonly found in T-C loop and D loops of tRNA molecules and explain why they are necessary. If there are 61 codons excluding the stop codons, then why are only 31 different tRNA molecules needed to carry out translation? How is a tRNA molecule charged with an amino acid? Compare and contrast ribosomes and the subunits in prokaryotes and eukaryotes. Right? What role does 23S-RNA play in synthesis of protein by a ribosome? And the 11th question, why is the correct reading frame important in translating an mRNA into protein? 12. Which mutation is more detrimental for an OR insertion of one extra nucleotide, or insertion of 3 extra nucleotides, and why? Summarize the initiation of translation in prokaryotes. Include the following terms: initiator tRNA, end-formal methionine, Shine-Delgado sequence, 16S-rRNA, AUG start codon, and initiation factors. What other codon is sometimes used as a start codon instead of AUG? What types of proteins tend to use this codon? Summarize, it is number 15, summarize the elongation step of translation in prokaryotes, including the following terms: E site, P site, A site, peptide transferase, translocation, elongation factors, and GTP. What are the 3 stop codons? What proteins recognize them? What is a polysome? Is number 17. Number 18, what is polycistronic mRNA? Is it found in prokaryotes, eukaryotes, or both? How does a prokaryote recognize an mRNA that needs to be translated? How about a eukaryote? What is meant by coupled transcription and translation? Does this occur in prokaryotes, eukaryotes, or both? Explain. What is tmRNA? What role does it play during translation? How does tmRNA tag proteins for degradation? In what ways is translation in eukaryotes simpler than translation in prokaryotes? Summarize initiation of translation in eukaryotes. In what ways is it different from prokaryotes? Number 25, how does the halting of protein synthesis due to nutrient deprivation differ between prokaryotes and eukaryotes? What is a signal sequence? What purpose does it serve? Summarize the process of co-translational export. What purposes do chaperone proteins serve? Number 29, compare and contrast bacterial protein export with mitochondrial and chloroplast protein import. Number 30, provide an example of how some organisms utilize mistranslation to create a functional problem. Number 31, why is the genetic code not completely universal? Number 32, why is post-translational modification? What is post-translational modification? Right, and number 33, why are selenocysteine and pyrrolysine considered the 21st and 22nd amino acids, whereas diphthomide is not considered the 23rd amino acid? How does a ribosome know to incorporate selenocysteine when it encounters a UGA stop codon? Which stop codon occasionally encodes pyrrolysine? Describe the mechanism of action of aminoglycoside antibiotics. Do you think spontaneous streptomycin resistance is likely to occur in bacteria? Why or why not? How does a cell protect itself from proteases that it normally exports after synthesis? What are lysosomes? How do eukaryotes dispose of unwanted proteins? Are the questions. There are 40 questions. Refer my lecture. Prepare your answers, and you can always post them on my comment section here on YouTube. I could verify them and clarify your doubts, right? You can also send your queries to my WhatsApp link as well. And you have some further reading. If you require further reading, you hear other references. Refer to the books Agros PF et al. 2017, Esnani M. Kumar P. Helen CUD 2015, Widespread Distribution and Structural Diversity of Type 4 IRESS in Members of Pycnobacteridae Virology, 478 pages, 6174. Page number 478, 6174. The first one by Agros PF et al. In 2017 is The Importance of Being Modified, the Role of RNA Modification in Translation Fidelity, the Enzyme 41. And third one be Ducler M. et al. 2016, Nucleoside Modification, this regulation of gene expression, focus on tRNA. And the next is Kun CD et al. 2015 on enzyme refolding permits, small RNA and tRNA surveillance by the CCA adding enzyme. Manso-CL, SC et al. 2016, bacterial abscess formation, which is controlled by the stringent stress response and can be targeted therapeutically, e-biomedicine. Wall, ML Louis, SM, 2017, methyl arginines within the RGG motif region of HNRNP-A1 affect its IRES transacting factor activity and are required for HNRNP-A1 stress granule localization formation, JMO. Werner F. 2012. A nexus for gene expression, molecular mechanisms of SBT5 and NUSG in the three domains of life. Willows J.E. tRNAs marked with CCA are targeted for degradations. And Yamashita S. Tomita K. about mechanisms of 3' mature tRNA discriminations from 3' immature tRNA by class 2 CCA, adding enzyme structure 24. Right? These are to do with references. Right? Right, we are proceeding with some Q&A. Now, protein synthesis translation is the production of a polymer of a chain of amino acids which produces a functioning protein. It involves reading the information from mRNA messenger RNA to put together a chain of amino acids. Ribosomes are the structures that synthesize the protein chain. Okay. As regularly as we see, people also ask, what cell is called protein synthesis? Which cell is I don't think the question is right, but anyhow, the answer is pretty fine. Ribosomes are the sites in which, in a cell, ribosomes are the sites in a cell which protein synthesis takes place. Okay, where in the cell does protein synthesis occur should be the question there. Anyway, cells have many ribosomes, and the exact number depends on how active a particular cell is in synthesizing proteins. For example, rapidly growing cells usually have a large number of ribosomes. Next would be, what is the purpose of protein synthesis? Okay, the purpose of protein synthesis is to make proteins for the cell and for the body. See, once this question Q&A is over, this lecture will be over, and then new one starts after this, all right? Proteins are important for carrying out chemical reactions, creating structures, acting as signaling molecules, and more. What are the most important steps in protein synthesis? The key step called translocation occurs after the bond is formed, joining a new amino acid to the growing protein chain. What is the mechanism of protein synthesis? The fundamental reaction of protein synthesis is the formation